The final category of organic macromolecules from Chapter 3 are the nucleic acids. Nucleic acids as a group are designed to store and transmit hereditary information. And we can think of the word hereditary meaning that it's going to be passed from generation to generation. Not always does this passing of information from generation to generation include from an individual organism to another. It could also be passing information from one generation of cells to the other. But we'll talk more about that later. So hereditary information is really important because it controls the activities of all of our cells. And if we look at how this is related to proteins, primary structure of a protein is determined by the sequence of the amino acids. The sequence of amino acids is ultimately going to be programmed by units of inheritance called genes. And these genes are sections of molecules of deoxyribonucleic acid, or DNA, that make up this hereditary information. So, nucleic acid examples include deoxyribonucleic acid or ribonucleic acid, RNA. Now, if we look at nucleic acids as polymers, we remember that they're put together using the dehydration synthesis reactions, and the monomers of nucleic acids are going to be called nucleotides. A nucleotide has three parts. There will be a nitrogenous base, either adenine, cytosine, thymine, guanine, or uracil. And you probably recognize these better by their first letter abbreviations, A, T, C, G, and U. So these nitrogenous bases are stuck onto a 5-carbon sugar, or a pentose sugar. Now the pentose sugar type will depend on whether we're talking about a molecule of DNA or RNA, because in a molecule of RNA, the 5-carbon sugar is ribose. In a molecule of DNA, the pentose sugar is deoxyribose. And then the final part of our nucleic acid is this phosphate, right? This phosphate group. So if we look at how we put these together, the nucleotides are joined by the bonding of the phosphate group from one nucleotide to the pentose sugar from the next nucleotide. And this forms something called a phosphate sugar backbone. Now, the phosphate sugar backbone is going to be formed by what are called phosphodiester bonds. So if we look at this graphically, here's our phosphate from one nucleotide, and it's going to be joined to the number 5 carbon of an adjacent nucleotide. And what this does is it gives our chain of nucleotides direction, because one end of the nucleotide is going to have a open 5' carbon, and the other chain of the nucleotides is going to have an open 3' carbon. Now let's go back for a minute and see where these come from. If we look at our nucleotide and our 5-carbon sugar, we're going to start numbering the carbons here. This is the number 1 carbon, the number 2 carbon, number 3, number 4, and number 5. So an adjacent nucleotide would have the phosphodiester bond occur between the number 5 carbon and this number 3 carbon. And so you can see how this forms this sugar phosphate backbone. The nitrogenous bases, A, C, T, etc., all kind of orient towards the side. And in a molecule of DNA, hydrogen bonding is going to occur between these base pairs, and we'll talk about that in just a second. So remember, these chains of nucleotides are directional because they have a 3' and a 5' end. And this is all related to which carbon is there. Okay? Now, when we look at the structure of DNA, DNA is composed of 2 complementary strands of nucleotides. And what the word complementary refers to is the information in one strand will determine the sequence of nucleotides in another, but they are not identical. And these strands, these 2 complementary strands of nucleotides in DNA, are going to be formed by hydrogen bonding of these complementary base pairs. And of course, the base pair rules: adenine forms hydrogen bonds with thymine, and guanine forms hydrogen bonds with cytosine. And this gives our DNA molecule a very unique appearance. It's often called the double helix. It's sort of like a double twisted ladder. And the two scientists that are given the credit for discovering the shape of our DNA molecule are Watson and Crick. And later on in the semester, when we get to talking about the actual discovery of the shape of DNA towards the end of this course, we'll talk a little bit more about Watson and Crick and their work winning the Nobel Prize for the discovery of DNA structure, but they were aided heavily by one of the most important woman scientists of our time, Rosalind Franklin. Now, the structured DNA not only allows for the DNA molecule to store genetic information based upon the sequence of these nucleotides, but it also allows for the DNA molecule to make a copy of itself, and we call this self-replication. So if a cell is going to divide and become two identical cells, making a copy of the genetic information is really important. And so DNA shape allows for it to not only store genetic information, but to go through this process called DNA replication or self-replication, where it makes a copy of itself. One of the things to remember, again, is that these strands are directional with the 3' and 5' ends. The strands of DNA, the complementary strands, also run in opposite directions of one another, and this is called anti-parallel. So if we want to look at an example of DNA replication, okay, here's a strand of DNA with a 5' end and a 3' end and a sequence of nucleotides, A, A, T, G, C, T. So which strand below, strand A or strand B, is the correct replicate for this section of DNA? So I'm going to give you a few seconds to think about this. And as you're thinking, I'll remind you that this is another one of these things that is really important. You being able to look at a sequence, a strand of DNA, and come up with its correct complement. So important that I'm going to put a question like this on the first exam. So, which one of these strands, A or B, is the correct answer? Most of you probably chose A because it's really easy to see the T and A, the G and the C together. But notice the 5 and the 3. Remember that this original strand's complement will run in the opposite direction. So the 3' end would pair here. T, T, A, C, G, A. Does everybody see why B is correct? All right, now RNA is a single-stranded molecule that's going to be made from our DNA template. One of the differences is that it contains the 5-carbon sugar ribose, not deoxyribose, and it will have uracil that replaces thymine. So uracil forms a hydrogen bond with adenine as we make our RNA templates. Now, as we'll see later in the semester, there's going to be several types of RNA, such as messenger RNA and transfer RNA, and these are all really important as we build our polypeptides. So, you should also be able to make the correct messenger RNA strand from this section of a DNA template. So here's our DNA template. Here's our RNA. You should be able to fill in these blanks. Notice that it's running in its anti-parallel fashion, so this would be thymine, right? No, this would be uracil, because remember, U replaces T, so we would have U, U, A, C, G, A. Now finally, what does all this mean? Well, we're going to talk more about this at the end of the semester, but basically if we look at a typical eukaryotic cell, we would see that our DNA molecules are inside of the nucleus. So here's our DNA molecules forming our inherited material, our chromosomes. And recall that organized on these chromosomes are genes or sections of DNA that are going to code for the building of a protein. So, in a nutshell, we're going to make a messenger RNA copy like this red strand. This represents our messenger RNA. We make a messenger RNA copy of our gene, and this leaves the nucleus and gets out into the cytoplasm where it finds an organelle called a ribosome. and the ribosome sandwiches between the messenger RNA sandwiches between the ribosome, and the information is read and translated. And individual amino acids, these little spheres here, are added in the correct sequence to make our primary structure of our polypeptide, and then ultimately secondary structure, tertiary structure, etc. So we'll spend a lot more time at the end of the semester talking about how DNA and RNA actually go through the process to build all of these proteins and ultimately control the way cells work.